Microfluidics enables the precise encapsulation of active agents, cells, or nanoparticles into droplets, microgels, or capsules. These encapsulation techniques use droplet-based microfluidic devices to create uniform particles with controlled size, shape, and shell thickness. Our semi-permeable shells provide protection against environmental stresses while enabling the controlled release of the encapsulated materials. Encapsulation can also enhance the stability and bioavailability of active agents by reducing degradation and prolonging their shelf life. Moreover, microfluidic encapsulation ensures uniform loading efficiency, which is critical for pharmaceutical applications and precision therapies. Advanced materials such as biodegradable polymers and smart hydrogels further enhance functionality, allowing responses to specific environmental cues like pH, temperature, or light. Encapsulation of mesenchymal stem cells in alginate hydrogels prolongs their viability and functionality, improving immune tolerance during therapeutic applications. Similarly, encapsulated enzymes maintain activity in extreme environmental conditions, broadening their industrial applications. Encapsulation via microfluidics is like wrapping fragile items in custom-engineered bubble wrap. It shields delicate materials while allowing access or release under specific conditions. Scaling microfluidic processes from laboratory research to industrial-scale production is a significant challenge, often addressed by parallelization and continuous flow designs. By integrating multiplex channels, microfluidic systems achieve high-throughput synthesis without compromising material quality or uniformity. Additionally, advancements in chip design, including multi-layered devices and modular systems, allow flexible and scalable setups. Continuous flow microfluidics ensures consistent production by maintaining precise control over reaction kinetics, fluid dynamics, and temperature. The use of automation and real-time monitoring further enhances scalability, enabling mass production with minimal waste. Such systems are particularly effective for producing functional polymers, nanoparticles, and drug formulations. Continuous preparation of calcium alginate fibers for tissue scaffolding avoids the traditional coagulation bath, enhancing efficiency and reducing labor. Scaling microfluidic production is like transforming a single artist's studio into a factory with robotic precision, maintaining artistic quality while significantly increasing output. Droplet-based microfluidics allows the creation of highly uniform droplets which act as microreactors for controlled material synthesis. These droplets enable precise control over reaction parameters, including reagent mixing, temperature gradients, and residence time, leading to high reproducibility and efficiency. Droplets serve as templates for synthesizing complex materials like particles, capsules, and hierarchical structures with unique optical, mechanical, or chemical properties. The droplet environment minimizes external contamination and ensures efficient reaction kinetics. Advances in surfactant chemistry have further improved droplet stability, allowing extended processing times for intricate material designs. Quadruple emulsions used to synthesize multilamellar phospholipid vesicles provide controlled drug release profiles for precision medicine. Droplet synthesis is like baking muffins in identical molds, where each mold ensures uniformity in shape, size, and texture. Double and triple emulsions, like water in oil in water systems, are advanced constructs enabled by microfluidic technology. These emulsions encapsulate both hydrophilic and hydrophobic substances, offering versatility for diverse applications, including drug delivery, food technology, and cosmetics. By fine-tuning flow rates and fluid properties, microfluidic systems create highly stable and customizable emulsion templates. For instance, the use of immiscible liquids in precisely controlled microchannels ensures the formation of consistent multi-layered droplets. These systems allow the incorporation of functional additives, such as stabilizers or responsive polymers, to enhance performance under varying environmental conditions. Triple emulsions for intestine-targeted drug delivery encapsulate sensitive pharmaceuticals, protecting them from degradation in stomach acid while enabling release in the small intestine. Creating double and triple emulsions is like designing a series of protective nesting dolls, with each layer offering a distinct role in shielding or delivering its contents. Stimuli-responsive microcapsules release their contents in response to specific environmental triggers, such as the pH, temperature, light, or mechanical stress. This functionality is achieved by incorporating responsive polymers or hydrogel materials into the capsule shells. These smart materials undergo reversible or irreversible changes in their structure, allowing precise control over the timing and location of release. Advances in stimuli-responsive materials include thermosensitive hydrogels, photoresponsive polymers, and magnetically activated nanoparticles. Applications range from controlled drug delivery to responsive coatings for smart textiles. UV-triggered photodegradable hydrogel capsules allow selective cell release in high-throughput cell sorting systems. Similarly, pH-sensitive capsules deliver anti-cancer drugs directly to tumor microenvironments where acidity triggers drug release. Stimuli-responsive microcapsules are like smart safes that open only under specific conditions, ensuring security and precision. Microfluidics offers unparalleled precision in the synthesis of nanoparticles, enabling control over particle size, shape, and composition. By managing reaction parameters such as flow rates, concentration gradients, and mixing efficiency, microfluidic systems produce nanoparticles with high uniformity and reproducibility. These characteristics are crucial for applications in drug delivery, imaging, and catalysis. Furthermore, microfluidic reactors can incorporate functionalization steps such as surface modifications with targeting ligands or stabilizing agents during synthesis. This integrated approach reduces the need for post-processing and enhances nanoparticle efficacy. Uniform gold nanoparticles synthesized using microfluidics exhibit enhanced optical properties for biosensing applications. Similarly, nanoparticles with tunable morphologies improve the efficacy of photothermal cancer therapies. Microfluidic nanoparticle synthesis is like crafting precision-engineered nanogems, where each particle is tailored for optimal performance. Hydrogels are highly porous, semi-permeable materials ideal for encapsulating cells, drugs, or biomolecules. Microfluidic systems enable the production of hydrogel microcapsules with tunable mechanical properties, pore sizes, and degradation rates. These parameters are essential for applications in drug delivery, tissue engineering, and regenerative medicine. Advances in cross-linking techniques such as photopolymerization and ionic gelation allow the customization of hydrogel properties to suit specific applications. Additionally, gradients in hydrogel stiffness or composition can be incorporated to study complex biological interactions, such as cell migration or differentiation. Hydrogel capsules with stiffness gradients are used to investigate cell behavior in mimicking tissue interfaces, such as tendon to bone junctions. Hydrogel microcapsules are like flexible sponges designed to hold and release their contents in a controlled and predictable manner. Microfluidics enables the synthesis of hierarchical photocatalysts with enhanced structural and compositional precision. By controlling particle size, porosity, and phase composition, microfluidic systems optimize photocatalytic properties such as light absorption, charge transport, and surface reactivity. Photocatalysts synthesized in this manner are used in applications ranging from environmental cleanup to renewable energy production. For example, microfluidic reactors can integrate dopants or create heterostructures to enhance the photocatalytic performance of titanium dioxide under visible light. Ternary composite titanium dioxide photocatalysts synthesized using microfluidics demonstrate superior pollutant degradation rates compared to traditional methods. Designing photocatalysts using microfluidics is like a jeweler cutting diamonds. Each structural detail enhances its brilliance and utility. Microfluidic systems facilitate the synthesis of functional polymers with specific architectures by controlling flow patterns, reaction kinetics, and monomer concentrations. These polymers are tailored for applications in coatings, adhesives, biomedical devices, and electronics. Advanced polymerization techniques, such as living polymerization and step-growth polymerization, are seamlessly integrated into microfluidic setups to produce highly uniform and functional materials. The ability to create block copolymers or surface functionalized polymers further broadens the scope of applications. Microfluidic systems enable the rapid assembly of multi-layer polyelectrolyte capsules, which are used for targeted drug delivery and separation technologies. Functional polymer synthesis in microfluidics is like weaving intricate fabrics, where each thread and layer is designed for specific functions and aesthetics. Inspired by natural structures, microfluidics is used to produce biomimetic materials that replicate the mechanical, chemical, or biological properties of living systems. By precisely manipulating fluid flow and material deposition, microfluidics enables the synthesis of materials that mimic the hierarchical organization found in natural structures like sea urchin spines, spider silk, or bone. These biomimetic materials exhibit remarkable properties such as high strength, flexibility, or biocompatibility, making them suitable for applications in tissue engineering, bioelectronics, and advanced materials science. Advances in three-dimensional printing integrated with microfluidics further enhance the fabrication of complex biomimetic architectures. Shear-aligned hydrogels mimic the ordered collagen structure of connective tissues, improving their mechanical properties and enabling use in flexible bioelectronics. Biomimetic materials are like artificial flowers, carefully crafted to replicate nature's beauty and function with precision. Janus particles and capsules feature anisotropic or directionally distinct structures, often with two or more regions possessing different chemical or physical properties. Microfluidic systems enable their production by leveraging immiscible fluid streams or precisely controlled polymerization processes at interfaces. These materials offer unique functionalities such as dual responsiveness to stimuli, self-assembly capabilities, and directional interaction. Janus particles are extensively used in targeted drug delivery, microactuation, and sensing applications, where their dual properties enable selective interactions. Advances in flow control and surfactant chemistry allow for the production of Janus materials with tailored surface chemistries. Stimuli-responsive Janus capsules release drugs selectively when exposed to specific triggers such as heat or light, while maintaining their integrity in other environments. Janus particles are like two-sided coins, with each side meticulously designed for a specific function or interaction. Microfluidic devices enable the production of highly stable emulsions with controlled droplet size and distribution. By precisely managing the flow rates and ratios of immiscible liquids, microfluidic systems produce emulsions that enhance texture, stability, and the controlled release of active ingredients. These emulsions are widely used in food technology to improve the sensory qualities of products like edible oils and sauces, and in cosmetics to stabilize lotions and creams. Microfluidics also allows for encapsulating bioactive compounds, preserving their efficacy until consumption or application. Edible microcapsules protect functional substances such as probiotics or vitamins from degradation during digestion, releasing them in the intestine for optimal absorption. Microfluidic emulsions are like perfectly mixed cake batters, ensuring even distribution of ingredients for the best outcome. Microfluidic-based encapsulation provides a protective environment for therapeutic cells, shielding them from immune attacks while preserving their viability and function. The encapsulating materials, such as hydrogels, can be engineered to exhibit optimal permeability, biocompatibility, and degradation properties, enabling controlled release and interaction with the host environment. These technologies enhance the efficacy of cell-based therapies, such as stem cell treatments, by extending the lifespan and functionality of the cells after transplantation. Mesenchymal stem cells encapsulated in hydrogels improve their therapeutic potential for tissue regeneration by protecting them from oxidative stress and immune reactions. Encapsulation for cell therapies is like placing delicate seedlings in a greenhouse, shielding them from harsh conditions while allowing them to grow. Microfluidic systems optimize the synthesis of lipid nanoparticles, crucial for delivering genetic materials such as mRNA in vaccines. These devices provide precise control over lipid composition, particle size, and encapsulation efficiency, enhancing the stability and efficacy of the nanoparticles. The uniformity achieved through microfluidic methods minimizes side effects and maximizes cellular uptake. Advances in automation and real-time monitoring enable scalable and reproducible production, meeting the high demand for vaccines during pandemics. Microfluidic platforms were instrumental in producing lipid nanoparticles for the COVID-19 vaccines, ensuring consistent quality and performance. Lipid nanoparticles are like armored vehicles, safely delivering delicate cargo, mRNA, to their destination with precision. Microfluidics enables the controlled synthesis of nanomaterials with tailored optical, electrical, and chemical properties ideal for sensing applications. By fine-tuning reaction conditions and material composition, these systems produce nanostructures with enhanced sensitivity and specificity. Such materials are essential for detecting chemical or biological signals in complex environments. Functionalization of these nanomaterials with ligands or coatings further enhances their ability to interact selectively with target analytes. Functional nanomaterials produced in microfluidics are used in artificial olfactory sensors, enabling the analysis of gaseous mixtures in industrial and healthcare settings. Nanomaterials for sensing are like highly trained sniffer dogs, capable of detecting and identifying specific signals with remarkable precision. Biodegradable microcapsules synthesized in microfluidic systems use natural or synthetic polymers that degrade under specific environmental conditions. These capsules ensure the effective delivery of active agents such as biocides or fertilizers, while reducing long-term waste and pollution. The biodegradability of these systems can be fine-tuned by modifying polymer composition, cross-linking density, or degradation triggers such as pH and temperature. This technology is crucial for sustainable applications in agriculture, medicine, and consumer products. Biodegradable microcapsules containing pH-responsive polymers release biocides in biodiesel storage tanks to prevent microbial contamination. Biodegradable capsules are like dissolvable packaging for detergents, disappearing once their job is done. Microfluidic systems produce vesicles which mimic biological membranes with precise control over size, composition, and encapsulation efficiency. These vesicles, including liposomes and polymerasomes, are widely used in drug delivery, diagnostic imaging, and biomimetic studies. Microfluidic production ensures uniformity, enhances stability, and enables the encapsulation of multiple components for targeted delivery. Advanced designs integrate multiple functions such as stimuli-responsive release and imaging capabilities within a single vesicle. Multilamellar vesicles, synthesized using microfluidics, enable the study of biomembrane dynamics and the targeted delivery of anti-cancer drugs. Microfluidic vesicles are like tiny balloons, carefully engineered to carry and deliver their cargo safely. Microfluidic synthesis enables the creation of hierarchical structures with multi-scale porosity and functional properties. These structures, which combine nano and microscale features, enhance performance in applications such as energy storage, catalysis, and drug delivery. Hierarchical designs improve surface area, transport properties, and structural stability. By controlling flow patterns and reaction conditions, microfluidics achieves precise layering, interconnectivity, and material integration. TiO2 hierarchical structures, integrated with CDS nanoparticles, enhance light harvesting and charge separation for efficient photocatalysis. Hierarchical structures are like multi-layered cakes, with each layer meticulously designed to serve a specific purpose. Asymmetric capsules, produced through microfluidics, feature distinct inner and outer shells, enabling multifunctional applications such as protection, targeted delivery, and controlled release. The inner layer often houses sensitive materials, while the outer layer provides mechanical support or selective permeability. By tailoring flow conditions and material properties, microfluidic techniques achieve precise shell asymmetry, enhancing capsule functionality. These capsules are particularly useful in mimicking natural systems such as extracellular vesicles for drug delivery and diagnostics. Advances in multi-phase microfluidics allow the creation of asymmetric capsules with gradients in stiffness, porosity, or responsiveness. Asymmetric capsules mimic extracellular vesicles to deliver nucleic acids efficiently in cancer therapies. Asymmetric capsules are like layered chocolates, with each layer serving a distinct function from protection to release. Microfluidics facilitates the integration of organic and inorganic components into hybrid materials, offering multifunctional properties such as flexibility, conductivity, and thermal stability. By precisely controlling reaction kinetics and fluid interactions, microfluidics enables the uniform assembly of hybrid structures at the nano and microscale. These materials are used in energy storage, catalysis, and advanced coatings. The incorporation of inorganic nanoparticles into organic matrices results in materials with enhanced mechanical and thermal properties. Hybrid microcapsules combining biodegradable polymers and inorganic nanoparticles improve durability and thermal stability in smart textiles. Hybrid material synthesis is like weaving silk with steel, combining flexibility with strength for versatile applications. Dynamic light scattering, or DLS, is integrated into microfluidic systems to monitor the size, distribution, and aggregation of nanoparticles or droplets in real time. This technique provides immediate feedback on reaction conditions, enabling researchers to optimize particle synthesis and ensure reproducibility. Microfluidic platforms with inline DLS reduce variability and enhance scalability in processes such as lipid nanoparticle production for vaccines or targeted drug delivery. Real-time data acquisition minimizes waste and allows fine-tuning of reaction parameters. DLS is used during the synthesis of lipid nanoparticles for mRNA vaccines, ensuring uniformity and consistency across batches. Integrating DLS in microfluidics is like using a thermometer while cooking, ensuring precise results every time. Microfluidics enables the design of capsules and particles with tunable shell properties for controlled release applications. By incorporating stimuli-responsive materials such as pH-sensitive hydrogels or temperature-responsive polymers, microfluidic systems allow the timed or localized release of encapsulated agents. These systems are critical in fields such as agriculture, where fertilizers or pesticides need to be released at specific rates, or in pharmaceuticals, where drugs must be delivered precisely to the target site. pH-sensitive microcapsules release fatty amines to combat corrosion in marine engines when acidic conditions are detected. Controlled release systems are like time-release capsules, ensuring delivery at the right moment and place. Microfluidics enables the creation of uniquely barcoded droplets, each acting as an independent microreactor for high-throughput screening. These droplets contain unique identifiers such as DNA sequences or fluorescent markers, allowing simultaneous analysis of thousands of samples. This technology is widely used in drug discovery, single-cell genomics, and molecular diagnostics. Barcoding ensures that each droplet's contents can be traced and analyzed, even in highly complex systems. Barcoded droplets enable simultaneous sequencing of thousands of genes during single-cell RNA sequencing experiments. Barcoded droplets are like assigning unique tags to packages in a warehouse, ensuring precise identification and tracking. Thermoresponsive microparticles produced using microfluidics change their physical properties such as size, permeability, or shape in response to temperature changes. These materials are designed for applications in drug delivery, tissue engineering, and smart materials, incorporating thermosensitive polymers such as poly-N-isopropyl-acrylamide enables reversible transitions between hydrophilic and hydrophobic states. This allows precise control over the release of encapsulated drugs or other active agents based on temperature fluctuations. Thermoresponsive microparticles release anti-inflammatory drugs at body temperature, ensuring targeted and controlled therapy. Thermoresponsive microparticles are like self-adjusting thermostats, adapting to environmental conditions. Metal organic frameworks, MOFs, are highly porous crystalline materials synthesized in microfluidics for precise control over their growth and properties. Microfluidic platforms enable uniform mixing of precursors and fine control over reaction conditions, resulting in MOFs with tailored pore sizes, compositions, and functionalities. These materials are used in gas storage, drug delivery, and catalysis due to their exceptional surface areas and tunable properties. Microfluidic synthesis eliminates batch-to-batch variability, enhancing scalability. MOFs synthesized via microfluidics are integrated into sensors for detecting volatile organic compounds with high sensitivity. MOFs are like sponges with precisely sized pores, optimized for capturing specific molecules. Biocompatible surfactants stabilize droplets in microfluidic systems, ensuring compatibility with biological samples and preventing cross-contamination. These surfactants maintain stable interfaces for reactions while supporting cell viability and biochemical activity. Applications include single-cell analysis, enzyme assays, and digital PCR. Advances in amphiphilic molecule design allow these surfactants to minimize toxicity and interference with biological processes. Amphiphilic polyphenol-based surfactants stabilize droplets in high-throughput digital PCR systems. Biocompatible surfactants are like a protective coat, ensuring stability while being compatible with delicate biological contents. Inspired by natural extracellular matrices, microfluidics creates three-dimensional hydrogels with tunable mechanical and biochemical properties. These hydrogels support cell growth, differentiation, and tissue regeneration. By controlling hydrogel network formation and incorporating bioactive molecules, microfluidics enables the production of scaffolds tailored for specific biomedical applications. Self-healing and stimuli-responsive hydrogels further expand their utility in dynamic biological environment. Polysaccharide-based hydrogels with ordered structures support slow drug release and enhance wound healing. Bio-inspired hydrogels are like custom-built scaffolds, promoting growth and repair in biological systems. Microfluidic flow reactors offer continuous platforms for material synthesis, allowing precise control over reaction kinetics, temperature, and mixing. These reactors enable efficient production of nanoparticles, polymers, and other advanced materials with scalability for industrial applications. The laminar flow characteristic of microfluidics ensures minimal waste and high reproducibility. Sequential or multi-step reactors further enhance complexity and functionality in synthesized materials. Microfluidic flow reactors synthesize gold nanoparticles with uniform size for biomedical imaging applications. A microfluidic flow reactor is like a conveyor belt in a factory, ensuring smooth and consistent production. Microfluidics creates advanced biomaterials and encapsulated cells for regenerative medicine. By mimicking native tissue properties, these materials improve cell viability, integration, and repair in damaged tissues. Techniques such as gradient hydrogel formation and cell encapsulation allow for precise control of mechanical and biochemical environments, promoting regeneration. These innovations are critical for applications in bone, cartilage, and neural tissue engineering. Hydrogel scaffolds with mechanical stiffness gradients are used to enhance tendon bone regeneration in orthopedic therapies. Microfluidic materials for regenerative medicine are like tailored prosthetics, designed to fit and function seamlessly in the body. Microfluidic systems produce materials tailored for environmental remediation, such as biodegradable capsules for controlled release of biocides, catalysts for water purification, and sensors for pollutant detection. By precisely controlling material composition and release kinetics, microfluidics addresses challenges like pollution control and resource efficiency. These materials often combine sustainability with high efficacy, making them critical for addressing global environmental issues. pH-responsive capsules release biocides at the water-oil interface in biodiesel storage tanks, preventing microbial contamination. Environmental microfluidic materials are like smart sprinklers, delivering just the right amount of resource where and when needed.